Hello, welcome to the research facility. So today I'm going to show you our research lab located in DRC at URMC. So this is our culture hood where we do our culture work, and we grow different type of cancer cell lines in T25 and T75's flask. And this is a scientific refrigerator, 2 to 8 degrees Celsius. Here we store all our media and prepared reagents and kits and this everything. And here next we have a centrifuge. So here we can centrifuge our sample to make a cell pellet. And here we have another culture hood. So in this culture room 2 person can work simultaneously. And then we have a water bath here which maintains 37 degree temperature, so that there are many reagents which are kept frozen. So before using that we need to thaw them, so we do that in this water bath. Then we have the inverted microscope here, under which we will put the flask. This is incubator. We have 2 incubators. So here, after culturing the cells in the flask, we put them into this incubator which maintains minus 37 degrees Celsius temperature, 37 degrees Celsius temperature and 5% CO2. And here we have a deep freezer which is minus 20 degrees Celsius. So here we keep all the reagents and kits which are supposed to be maintained at more than minus 10 and above. So whenever required, we just take out the reagent from this deep freeze and thaw it in this water bath. Here we have a microcentrifuge. So here we can centrifuge the little Eppendorf tubes. And I think that's all in this room. And let's move to another room. So here in this room, this is a storage room. Here we have nitrogen tank, one and other. In these tanks we store the cell lines in liquid nitrogen, which can be stored for many years. Similarly, we have minus 80 degree deep freezer. Here we store the cell lines in the boxes, which can be stored for a longer time. And then we have 4 degree big refrigerator in which we generally store our kits and media, culture media. Let's move to the next room. Okay, this is another culture room. Here we have a culture hood and the refrigerator. And here we have 3 incubators, 1, 2 and 3. And here we have 1 water bath, inverted microscope which we saw in the other room, and turn on the power. And the most important thing we have is this big compound microscope, which is a kind of combination of a light microscope as well as it has this immunofluorescence attachment, so you can examine the normal slides under light microscope as well as use immunofluorescence. It has a camera attachment through which you can transfer the images to this computer. Okay, so this is our main laboratory, and here is the RNA, DNA, and protein section. So with this kits, we do Western blot testing here, and here we have another centrifuge. Then here we have a refrigerator which is minus 2 to 8 degrees Celsius. We store the kits and reagents here as well. Particularly we store the reagent which we prepared in-house, which are not ready-made. And then this is the station for RNA isolation. This is another centrifuge in which you can adjust the temperature as well and speed. And here we have minus 20 degrees refrigerator. And these are our desks, seating desk, and yes, this is the machine we have here is Quantum Studio 3. It's a fully automated RT-PCR machine. This is another RT-PCR machine we have. It is a Bio-Rad, and yes, this is immunohistochemistry bench. So here we do immunohistochemistry experiment and prepare the IHC slides. This is the hot plate on which we warm the slides. And here we have one freeze which is 2 to 8 degrees Celsius, and along with the freezer which is minus 20, and this is standalone minus 20 degree deep freezer. That's it. This is our lab. Thank you very much for watching the video.